Okay, welcome everybody to a new global ImmunoTalk. I'm Carla Rothlin, and I'm here today with one of our co-organizers, Dr. Enrique Veiga Fernandez. I would just like to remind you that next week we'll have Dr. Pipa Marak, so we look forward to connecting with you again next week. And now I will let Enrique introduce our fabulous speaker today, Dr. Fiona Powrie. Thank you.
Thank you, Carla. It's really my enormous pleasure and great honor to introduce today's Global ImmunoTalk speaker, Fiona Powrie. I think it's one of those speakers, it goes without saying, that probably would not need any formal introductions. We all know Fiona's work and the enormous contributions she made to immunology and science in general. So Fiona is currently director of the Kennedy Institute of Rheumatology at the University of Oxford. She's also fellow of the Royal Society and governor of the Wellcome Trust. Fiona was actually born in the United Kingdom, where she graduated in biochemistry, and then she gained a PhD in immunology, also at the University of Oxford. And then she moved for a certain period, I think about 6 years, to the DNAX Research Institute, time in Palo Alto, before then returning back to Oxford. That was in 1996. And actually, when she came back to Oxford, she became a Wellcome Trust Senior Research Associate. During this period and before being the director and joining the Kennedy Institute of Rheumatology, Fiona was also a Sidney Truelove Professor of Gastroenterology and head of the Translational Gastroenterology Unit in Oxford. This from roughly 2009 to 2014. So Fiona's research actually has been absolutely pioneer and transformative in the understanding of the relationships that are established between the microbiome and the host immune system in the intestine, and also on the understanding of how these relationships break down in inflammatory bowel disease, but also in other diseases, including arthritis and cancer. Actually, in a recent work, to give you an example, she actually revealed and showed that a very important molecular axis that now all immunologists are acquainted with, the IL-23 Th17 axis, can actually act as a driver of colorectal cancer. So this is just one of the numerous contributions she made. She also has identified an important cytokine, OSM, as a biomarker of, and a therapeutic target in resistant IBD to TNF, anti-TNF treatment. So it comes without saying that all her contributions were then published in top-ranked journals with landmark papers, and it comes without saying that she received many accolades and prizes. I will just highlight two of them that I consider really exciting and very important. She actually was awarded the Louis Jonte Prize for Medicine in 2012, and she was elected more recently as an international member of the National Academy of Sciences in 2020. So Fiona, thank you so much for being here with us. Welcome. It's really a pleasure to have you on board for today's Global ImmunoTalk.
Enrique, Carla, thank you very much. One, for that very warm introduction. I'm feeling old when an introduction goes on for a bit. It means you've been around a while. But it's really inspirational to be part of this series that I believe is broadcast around the world. So hello to everyone. I hope you're enjoying these talks. I wish I could see you in person, but immunology, science is what is enabling us to defeat this horrid pandemic. So we're in the right place.
Absolutely. I mean, the introduction takes for so long is not because you've been around for too long. It's just that your work is sufficiently important to deserve a long introduction. So we normally have a question that allow the audience to both to get you to know a little bit better from a personal perspective, but also we like to inspire the new generation of scientists in general and immunologists in particular. So what I would like you to ask you today is, what would be the single most important advice that you would give to the new generation of immunologists that are on the other, or is on the other side of the screen attending this talk?
Yeah, thank you. I mean, wherever you are, I would encourage you, a career in research in immunology is an excellent opportunity. Believe in yourself, follow your ideas, be persistent, and take the opportunities when they arise. It's with this type of knowledge that we're able to improve health and contribute to better societies. So it's a very worthwhile thing to do.
Wonderful. Thank you, Fiona. So Fiona's talk will certainly address very important questions in the context of immunology. That's what we're doing here, and that's the main topic of this seminar series, and certainly will relate to mucosal immunology in the gut. And the title of our talk is Translating Immunology into New Therapies for Inflammatory Bowel Disease. Fiona, the stage is all yours, and I thank you very, very much. Thank you ever so much for joining us today, and we look forward to your exciting talk. Thank you.
Well, thanks again. I think Carla's going to pull up my slides. Is it now, Carla, or?
I think it will be great if you can start sharing your screen. I think it should work. Let's see. Perfect. Yeah, and if you can put it as presenter.
So I'm sharing. Could you?
Yes, you're sharing your screen. We see, and yeah, if you can press that, then perfect. That's in presenter mode is fantastic, and you have your laser point. We're ready to go. Thank you so much.
Fantastic. Well, thanks again for the introduction and for inviting me. So I want to tell you about our studies deciphering communication between our large and complex gut microbiota and the underlying host, and how we can harness that knowledge to inform the development of new therapies for inflammatory bowel disease. I would normally be working at the Kennedy Institute, which you can see here. When we went into lockdown, the Kennedy was 2 floors in height. And now that we've finished lockdown almost, we're getting there, we've built an additional floor. So that's an exciting horizon for the Kennedy Institute, whose work is focused on understanding chronic inflammatory diseases. Before going on, I just wanted to dedicate this lecture to Don Mason. Don sadly passed away earlier this year. Don's a very important person in my life, my PhD advisor. He was a really pioneering immunologist with some of the early work describing immune regulatory pathways, including the regulatory T-cell axis. It was really a privilege to work with Don as an outstanding scientist, but also as a wonderful human being, and he will be sadly missed by us all. So here is the gut. Here is the host microbe interface in the gut. The gut is a primary site of host microbiome symbiosis, and I always find it remarkable that such a large density of microbes stained here in yellow, picking up pan bacterial message, living so close to the epithelial cell surface. And of course, there are important interactions at this interface that I think are fundamental for health. And one of those is signals from this diverse microbiome composed of hundreds of different species, which is highly individual, but collectively these microbiomes encode a range of molecules and metabolites that induce the development and function of the immune system. And of course, that is important to maintain appropriate barrier, healthy barrier responses. through the production of mucins, antimicrobial peptides, and immunoglobulin A, amongst many of the pathways that are involved in maintaining healthy barrier responses. Now, this dialogue can break down in disease and is controlled by both genetic and environmental factors, and I'll be coming to some of those as we go through the talk. What I'll focus on today is a maladaptation leading to the development of inflammatory bowel disease, but a healthy gut barrier is relevant to health and disease across a range of different areas, as I've no doubt you've heard about from others during this series. Now, in inflammatory bowel disease, this is a very complex, multifactorial disease that has different clinical presentations. There are different clinical forms. Crohn's disease and ulcerative colitis are the main forms. They involve different location of the lesions, as well as different pathological features. But I think it would be fair to say that even within Crohn's disease or ulcerative colitis, there is great heterogeneity within IBD, with some patients having a fairly straightforward type of disease that can be treated by frontline immunosuppressants, whereas others will progress to requiring biologics or even surgery. It's a particularly insidious disease. It strikes young individuals in their prime, with the peak age of onset around 20 years of age. There's a high prevalence, which is increasing. And although therapies such as anti-TNF have been transformative, around half of the patients will either lose response or fail to respond to anti-TNF and develop down a route of more severe disease. And I think one of the biggest challenges is that with the heterogeneity of disease, we have limited information on how to target treatments to the right patients. So as you'll see, we are getting an increasingly stocked toolbox to be able to treat inflammatory bowel disease, but we don't really understand how to apply that in a rational way to the different forms of disease. And undoubtedly, that is a consequence of this multifactorial nature of IBD, involving the interaction between genetic predisposition and environmental triggers that play out in this relationship in the microbiome and the host immune response. All is not gloom, though. I think we're making important strides in our understanding, and really the last decade has brought a number of new technologies and opportunities that allow us to link discovery and translational research in inflammatory bowel disease. There are an increasing number of animal models of disease, and of course there's an increasing ability to be able to understand human immunology, cohorts of longitudinal cohorts of IBD patients, understanding about the polygenic disease, 230 genetic loci that have been linked with inflammatory bowel disease. And there are also important messages to be learnt from IBD-like Mendelian disorders. And these are, of course, extremely rare disorders involving single genes, often gene deletions. And they have really told us about key pathways that are required for intestinal homeostasis, and how when they are altered, this can lead to inflammatory bowel disease. It's also worth noting that with the advent of checkpoint blockade to enhance anti-tumor immunity and treat and hopefully ultimately cure cancer by harnessing the immune system, an adverse event of this can be the development of colitis that occurs in around 30% of patients treated, for example, with anti-CTLA-4. So those unfortunate side of probably on-target effects offer an opportunity to study an inflammatory bowel disease like syndrome that develops with a defined onset following administration of these drugs. And of course, our ability now to be able to manipulate not only the microbiota, but also study intestinal epithelial cells using organoids to utilize induced pluripotent stem cells, as well as single cell technologies, really offer the opportunity in these model systems, as well as in human IBD patients, to start to get granularity about the cellular nature of disease and the molecular processes that underlie that. What we're understanding from the genetic studies as well as functional studies are that there are key, what we call pillars of intestinal homeostasis. One of those is the intestinal epithelium and how it is able to compartmentalize the microbiome. What we don't want, although the microbiome provides a lot of positive signals, we don't want it penetrating through the epithelium and into the underlying lamina propria. And there are a number of barrier defense mechanisms involved in that, and genetic deletions in genetic alterations in those can result in Mendelian forms of inflammatory bowel disease. The other area that is a fundamental pillar is the ability of mononuclear phagocytes to take up bacteria and to be able to kill those bacteria. So if the barrier is breached, then there is a layer of innate immune defense, MMP driven microbicidal function that plays an important role. And again, deficiencies in this function can give rise to inflammatory bowel disease. And of course, the area that we have been particularly interested in is the function of regulatory T cells that control the inflammatory response, particularly those expressing FOXP3. And there are patients who have deletion of FOXP3 or a number of the downstream pathways associated with regulatory function, particularly interleukin 10. They have deficiencies in those pathways, will also develop a Mendelian inflammatory bowel disease. Again, highlighting the non-redundant role of the regulatory T cell axis in controlling microbial responses in the intestine. So our work initially started with various mouse models. This is the T cell transfer model. This is an extremely simplified model, but what it revealed to us is that the naive T cell repertoire has the capacity to mount a deranged response to the microbiome, and that is normally controlled by regulatory T cells. So in this model, transfer of naive T cells into mice that have no T cells of their own leads to the development, the differentiation of these cells in the intestine. Well, it leads to the accumulation of Th1 and Th17 cells in the intestine, and the development of an IL-23 dependent colitis. And in fact, that can be blocked by a number of antibodies against key mediators. And of course, this was in fact one of the first models in which anti-TNF was shown to inhibit colitis. And of course, that has been revolutionary in treating inflammatory bowel disease patients. What also emerged from these studies was the role of IL-23 in treating perhaps a distinct, in driving disease in a distinct subset of patients. What we found in the early studies was that transfer of regulatory T cells defined by the signature transcription factor FOXP3 into mice that either before they develop colitis or when they have established colitis is able to ameliorate disease, and that was involved a mechanism involving the elaboration of interleukin-10, and there's a little bit more about that here. Another model that we've used that allows us to look at the response to a particular bacterium is Helicobacter hepaticus infection, and I want to spend just a minute explaining this model because I'll be talking a bit more about that as we go through. Now, Helicobacter hepaticus is a gram-negative microaerophilic bacteria. Here in green, you can see it lives up close in the gel mucus layer, very close to the intestinal epithelium, in what Laura Hooper has described as the demilitarized zone. So Helicobacter is unusual in occupying that particular niche. Now, Helicobacter hepaticus will establish a lifelong infection in normal mice, C57 black 6 mice, for example, and it doesn't elicit immune pathology, although it does elicit a strong immune response, including IgG antibodies, but there's no immune pathology. And one of the reasons for that is Helicobacter elicits a very strong regulatory T cell response with an accumulation of FOXP3 expressing cells that can make IL-10 in the intestine. Now, if we cause a maladaptation between the host and Helicobacter hepaticus by neutralizing IL-10, rather like these monogenic diseases patients that don't have a functioning IL-10 axis, if we take IL-10 away by administering an anti-IL-10 receptor antibody, then these mice will develop intestinal inflammation, and it is driven by production of the cytokine interleukin-23, because if we negate IL-23 receptor signaling on T cells, then the mice do not develop intestinal inflammation. So there is a requirement for the production of IL-23 and IL-23 signaling for development of this disease. What's of interest about this model, and something we've used later, is that it doesn't respond to anti-TNF treatment. So it's an interesting model in the when thinking about the unmet clinical need of IBD patients that fail to respond to anti-TNF. And in this model, IL-23 appears to be important, but I'll talk to you about some other mediators that are involved. So these types of experiments have led us to conclude that an important balance in the intestine is the balance between the production of interleukin-10 and the regulatory T cell axis on one hand, and Helicobacter hepaticus in this model system is able to promote the development and functioning of this population of cells. But if that axis is disrupted by, for example, taking away IL-10 through the anti-IL-10 receptor treatment, then now Helicobacter hepaticus drives this inflammatory Th17 response with elaboration of interleukin 23. And I think this model of the balance between the regulatory T cell axis and the inflammatory Th17 axis is validated by the findings from Mendelian diseases with IBD that develop as a consequence of IL-10 and Treg deficiencies, and the fact that IL-23 illuminated in these and other studies, collaborations with Dan Qua and colleagues, has been shown to be effective in the clinic in inflammatory bowel disease. So what I want to talk about this afternoon with you is some of our newer data looking at the compartmentalization of the regulatory T cell response. Can we track antigen-specific regulatory T cells in the intestine? Where do they go and what do they do when they get there? That's very much work in progress. And another newer area, which is a focus on a pathway of myeloid mesenchymal activation that occurs in anti-TNF resistant IBD. So I'll explain a little bit more about that. And that is really taking our research into human IBD patients. So what I've told you so far is that Helicobacter hepaticus is able to induce this regulatory T cell population. This was just studying polyclonal T cell responses, and we've spent some time trying to characterize that activity a little further. Camille Dan, when she was in the lab, showed that Helicobacter hepaticus makes a polysaccharide that is able to bind to TLR2 and influences cytokine production by macrophages, promoting the IL-10 response whilst inhibiting pro-inflammatory cytokine production. And that pathway involved activation of the transcription factor CREB and activation of a range of repair pathways in those macrophages. So further work to understand precisely how this is functioning and whether this is we have speculated that this is a pathway of host microbial symbiosis. So of course, one of the things I mentioned was being able to track antigen-specific regulatory T cell responses, and that is another technology development in the area. We are very grateful to Dan Littman, who made a Helicobacter hepaticus reactive T cell receptor transgenic mouse. That's been an enormously useful tool to track T cell responses following Helicobacter colonization, and they published a very nice paper on this a couple of years ago. Emily Thornton and Yizu Gu. So Emily Thornton was a postdoctoral fellow in the lab, but now establishing her own lab, and was supervising Yizu Gu, a clinician scientist PhD student who has just written her thesis. And this is some of the work, which is really a work in progress, but they were using the TCR transgenic mouse model to transfer Helicobacter reactive T cells into naive mice that are challenged with Helicobacter hepaticus. The donor T cells express a different allele of CD45, so we can track them and look at what happens to them in different immune compartments. And as Dan had described, and from what we would have expected from the polyclonal responses we described much earlier, is that the Helicobacter reactive T cells, naive T cells, do differentiate into FOXP3 expressing cells, a rather large proportion of them become FOXP3 expressing cells and are able to produce interleukin 10. And this is data from the cecal patch. And what Emily and Yizu have been doing, and I'm just going to give you a snapshot of the approach, is to ask the question, in that setting where we transfer Helicobacter reactive cells, colonize with Helicobacter, what is happening to the T cell response as the cells are primed and start to infiltrate the intestine. And Yizu has been particularly interested in understanding the fate of the T cells in different compartments within the intestine, the cecum, where there is a large cecal patch and an organized lymphoid structure, to the lamina propria within the cecum and the colon, where there are smaller lymphoid aggregates. And what Yizu and Emily have found is that following transfer, and I think this is around day 7 following transfer, the highest proportion of FOXP3 expressing cells are found in the intestine, and that response is particularly in outside of the larger organized structures. So MLN has a lower frequency of FOXP3 expressing cells, as does the cecal patch, and these more larger organized lymphoid structures in the colon. So cells will be primed in the mesenteric lymph node, and then they are accumulating in the intestine, and there's a differential accumulation of regulatory T cells, particularly outside of these organized structures. And that's true not only of FOXP3 expression, but also of the ability of the T cells to produce interleukin 10, detected here using crossing the T cell receptor transgenic to a reporter strain for interleukin. Yizu and Emily have been doing some live cell imaging, and the point to make from this is that whilst the FOXP3 positive cells, they've differentiated into FOXP3 expressing cells from naive T cells, they must be encountering Helicobacter antigens, you get this strong induction of FOXP3 expressing cells. They are present in these small in the larger organized clusters, as well as in these smaller clusters in the intestine. But what's different is that the ability to produce interleukin 10 appears to be outside of these structures. So there's clearly a signal that is influencing regulatory T cell effector function in the tissue. And Emily and Yizu are employing niche-seq type approaches to be able to identify what regulatory T cells are interacting with in the lamina propria that influences the induction of interleukin 10. Just shows you here that IL-10 quantitatively is produced at much higher frequency of cells out in the lamina propria compared to in these small organized structures. So I think this is an exciting area to be able to track the fate of T cells responding to a pathobiont in the form of Helicobacter. These cells, as Dan has shown, are primed in the mesenteric lymph nodes. They then go into the intestine, and here there's a differential accumulation of regulatory T cells. Is that because there's an element of homing following priming, or some accumulation and proliferation here but there's a Treg enrichment in the gut. And furthermore, the ability of regulatory T cells to produce IL-10 is anatomically compartmentalized from those that are present in the lymphoid aggregates. These regulatory T cells are not in organized structures in the lamina propria where they're turning on interleukin 10. And it will be, of course, of interest to understand the bacterial signals as well as the host antigen presenting cells and stromal compartments that are involved in the compartmentalization of this response, possibly offering the opportunity to be able to intervene in that, to promote it in certain settings or to inhibit it in others. So I want to turn now to difficult to treat inflammatory bowel disease, to review some of our work involving IBD patients. And really this work started 5 or 6 years ago with an idea from 2 talented fellows in the lab, Ahmed Higatzi, a clinician scientist, and Nathan West, a basic scientist, who asked the question whether there were cytokines that we had missed that were associated with therapy-resistant IBD. They started by interrogating the range of publicly available data sets for Crohn's disease and ulcerative colitis patients. And they came up after looking at large numbers of patients in a range of different data sets. They came up with 4 cytokines that were consistently increased in both Crohn's disease and ulcerative colitis. Some were more specific for UC or Crohn's disease, but what I'd like to focus on are those that were consistently elevated in Crohn's disease and ulcerative colitis. They 3 of those were well known to us, interleukin 1, alpha and beta, and I will come back to interleukin 1, interleukin 6, and another member of the IL-6 family, oncostatin M. Now, I must say, when Nathan and Ahmed brought this data to me, I thought, what on earth is OSM? I don't know anything about OSM. I think it's been somewhat overlooked because it's it's not an interleukin, but it is an important cytokine from the IL-6 family. OSM is produced by leukocytes. Its receptor is on stromal cells, and it's been shown to function in hematopoiesis, liver repair, and skin and CNS inflammation. But at the time we were working on this, there was very little known about oncostatin M in the setting of the intestine or intestinal inflammation. The OSM receptor is, of course, a heterodimer with the common GP130 from IL-6 family receptors and the unique oncostatin M receptor. The work that Ahmed and Nathan did, and I don't have time to go through all of this with you, it was published 3 years ago now, showed that oncostatin M signals through receptor expression on intestinal stromal cells, including fibroblasts and endothelial cells. So I've just got a couple of slides to review that data and then to show you how we have built on that in the concept of, towards the concept of distinct pathotypes in inflammatory bowel disease. So the finding that OSM was consistently elevated in both Crohn's disease and ulcerative colitis led Nathan and Ahmed to ask whether it was associated with or predictive of non-response to anti-TNF therapy. So they assessed oncostatin M and expression of its receptor in cohorts of patients that were sampled just as they were going on to anti-TNF therapy. There were biopsies taken before, and then the response was monitored. And indeed, oncostatin M and its receptor were very strongly associated with non-response to anti-TNF in both Crohn's disease and ulcerative colitis. OSM had a very high sensitivity and specificity for identifying anti-TNF non-responders. But the expression of OSM and its receptor is not simply a biomarker, and of course the utility of that remains to be tested in a properly controlled prospective study. They also showed that oncostatin M plays a functional role in this anti-TNF refractory model of Helicobacter-driven colitis with IL-10 receptor blockade. And what you can see here is development of severe colitis in Helicobacter infected mice with anti-IL-10 receptor blockade, with the development of crypt abscesses, the accumulation of some neutrophils in mice that were genetically deficient in oncostatin M. They did develop colitis, but the severity and the type, the pathology was reduced. So disease was attenuated and the mice were able to recover earlier. And what was particularly lacking was the crypt abscesses and accumulation of neutrophils. So that study, together with a number of studies from others, from the Simmons lab, as well as Ramnik Xavier and others, have utilized single-cell sequencing to define the cellular landscape in IBD as well as in therapy-resistant IBD. And where we've got to from our own study, as well as these fantastic single-cell sequencing atlas style approaches, is that there is a cellular landscape in therapy-resistant IBD. There are inflammatory mononuclear phagocyte populations expressing oncostatin M, interleukin-1, as well as IL-23, as well as a number of different inflammatory fibroblast populations, as well as IgG plasma cells and activated T cells. And what we don't know here is the functional interactions and the tissue ecology in IBD in which they operate. And that's what I'd like to address with the final part of my seminar this afternoon. So there are clearly inflammatory pathways associated with therapy resistance in IBD, and we've wanted to further characterize those in terms of cellular interactions and the tissue niches in which they operate. And this is work from Matt Jackson, Matthias Frederick, Mathilde Pohin, and other colleagues in the lab. And they used a discovery cohort of patients undergoing surgical resection, some of them for inflammatory bowel disease, controls for using unaffected parts of tissue from those undergoing surgery for colorectal cancer. And what Matt, Matthias and Mathilde have done is at this snapshot of disease tissue, and of course often individuals going into surgical resection have therapy resistant disease, not all of them, but we have an excellent set of metadata on these patients to be able to correlate aspects of their disease with the gene expression pathways that are activated. So the approaches are, of course, whole tissue transcriptomes, single cell sequencing, but importantly, trying to map these in situ to particular tissue ecologies and niches. So with that large cohort there of IBD surgical resections, cases and controls from the whole tissue RNA sequencing, Matt performed a weighted gene co-expression network analysis, which actually takes all of the genes after the filtering process, some 22,000 genes, and clusters them on those into modules of co-expression. So those that are most closely clustered in terms of their expression, and then with those modules looks at their association with different aspects of the metadata. And there's a huge amount of information in here. This work is on bioRxiv for those who want to look at it in more detail. But I just want to draw your attention to this, to two modules of around 50 or 60 genes each that strongly correlate with the microscopic analysis of inflammation, the so-called Nancy index. This module also strongly correlates with macroscopic inflammation. So we were interested in further pursuing what genes were in this module and how did that map to cells in tissues. And I'll take you through that. But before that, because we're interested in therapy refractory disease, that's where there's a great clinical unmet need, Matt and colleagues asked the question whether expression of the genes in these modules was a predictor of anti-TNF non-response, or was associated with anti-TNF and corticosteroid non-response. And that is true. M4 and M5 were the most predictive or associated with therapy non-response. So what do these modules actually relate to? We can see the M4 and M5 genes listed here on the x-axis. What we've done here is sort different immune cell populations from the inflamed intestine and analyze the expression of these module genes within the different cell populations. And I think just looking at the colors, brown for a high level of expression, one can see that a large proportion, about half of the genes in M4 and M5 are contained within stromal cells, and that involves genes such as IL-11, podoplanin, neutrophil attracting chemokines, whereas the other half are expressed not in mononuclear phagocytes, as we might have predicted from the single cell sequencing data, but are enriched in neutrophils. And that includes oncostatin M itself, strongly enriched within the neutrophil, within neutrophils from the inflamed intestine. We wanted to map these modules, as I've mentioned to you, of gene expression that appear to relate to a activated stromal neutrophil axis to tissue pathology. So we went through the discovery cohort and the surgical resections, and Alistair Easton, a histopathologist, quantitated the different forms, histological features of inflammation. And the types of thing he was looking at was architectural distortion and goblet cell depletion, lymphoid aggregates, inflammation in the crypts, cryptitis, crypt abscesses with neutrophil infiltration, granulomatous inflammation, and ulceration. And there's rather a lot of data here, but what he was able to show was that M5 and M4 modules were related to ulceration, strongly correlated. Those genes, their expression was strongly correlated with ulceration in the tissue. By contrast, there were other modules, and M6 is one of them, and that has a much larger number of genes, so there's more work to be done on the M6 module, but that is strongly associated with lymphoid aggregates. So we're seeing linking gene expression to distinct types of tissue pathology that we're describing as IBD pathotypes. Alongside this work, in collaboration with colleagues in the Roche fibroblast network and Ilya Kozunski in particular, we have done some single cell sequencing of biopsy samples from inflammatory bowel disease patients, affected and unaffected tissue, as well as across a range of other inflammatory diseases. And there's a paper about that from Ilya and colleagues also on bioRxiv. But I want to talk about the changes that were specific to the intestine. And we were able to detect in that single cell sequencing, where we found a number of different subsets of stromal cells, endothelial cell populations, smooth muscle cells, pericytes, and a number of different fibroblast clusters, as have been described by others, a population of inflammatory fibroblasts, which have also been noted by other investigators. And what was striking about that population of inflammatory fibroblasts was the upregulation of a number of neutrophil attracting chemokines, as well as activation markers, fibroblast activation markers such as FAP and podoplanin. So a number of chemokines that promote, that recruit neutrophils through binding to CXCR2. And we can actually detect that interaction again at sites of ulceration between activated neutrophils and fibroblast populations in here, those activated fibroblasts in magenta, which express both podoplanin and FAP in close proximity in granulation tissue in these ulcers, with neutrophils expressing neutrophil elastase. So in this pathotype that involves an activation of fibroblasts, the accumulation of neutrophils, the production of oncostatin M by those neutrophils, what is driving the production of chemokines that recruit the neutrophils to the effect? And of course, there's been a lot of work done on cytokines that induce the production of neutrophil attracting chemokines. And these are some in vitro experiments utilizing a fibroblast target line and stimulation with IL-1 and TNF leads to the production of a number of different neutrophil attracting chemokines by fibroblast pop. So the sort of final piece of data was to show that in conditioned media from inflammatory bowel disease lesions, that conditioned media is able to also induce the production of neutrophil attracting chemokines, and that activity is inhibited by an anti-IL-1 signaling reagent, the IL-1 receptor antagonist anakinra, but not by an anti-TNF. So we conclude in this particular pathotype of inflammatory bowel disease, there is IL-1 driven activation of fibroblasts with the elaboration of neutrophil attracting chemokines and the accumulation of neutrophils at these sites of ulceration. And indeed, one can map precisely interleukin-1 expression, IL-1 beta, at these sites of ulceration in very close proximity to activated fibroblasts, as highlighted by staining with FAP. So in summary, identification of distinct pathotypes associated with treatment non-response in IBD, which raises the prospect of individualized targeting. A subset of treatment refractory IBD is associated with an activated neutrophil stromal cell axis, activated inflammatory fibroblasts producing neutrophil attracting chemokines map to ulcerated tissue, and IL-1 is an important upstream driver that induces fibroblast activation and maps to the site of ulceration. Importantly, activated neutrophils produce OSM, a previously unrecognized mediator of intestinal inflammation. And of course, this type of neutrophil fibroblast crosstalk drives tissue remodeling and pathology. And of course, what is the relevance of this to other inflammatory diseases? So what we think is happening in these patients that have treatment refractory IBD is that there is a disruption of the epithelial barrier, the penetration of microbial stimuli leading to the activation of local fibroblast populations that remodel this part of the tissue, the accumulation of neutrophils, and an attempt to wall off the barrier. So is this a deranged repair response? What are the relevant upstream drivers of interleukin-1? And there are a range of different contenders there, but importantly, will targeting of the neutrophil fibroblast axis be a new therapeutic strategy in treatment refractory IBD patients? And of course, the big picture here is to move towards not only gene modules that are elevated in different subsets of IBD patients, but to actually map those to pathotypes and distinct tissue ecologies, which may give us a way of targeting treatments to particular processes that drive the disease response in tissues. And I want to finish there then with thoughts towards a new process-driven taxonomy of disease, away from the individual diseases and their clinical symptoms to identify cells and pathways that may be shared between a number of different diseases that would be targeted by a particular mediator. So anti-IL-23, anti-oncostatin F, this neutrophil fibroblast type of interaction. This has been a huge effort. I've been so lucky to work with such outstanding students and postdoctoral fellows over the years. I've mentioned those who contributed to these studies in my lab, both current and former members as we went along. We've had excellent collaborations with Alistair Easton in histopathology, Holm Ulig and colleagues in the Oxford TGU and Biobank, the Roche Fibroblast Network, as well as colleagues at Janssen R&D in the early OSM studies. And of course, I'd like to recognise the financial support of our sponsors. Thank you very much.
So Fiona, thank you so much for this extraordinary, absolutely inspiring talk, not only from published data, but also in progress work in the lab, revealing this complexity of landscapes in IBD patients. And I think this is a very strong message to the young audience out there that inflammatory disease is not only about immune cells, and you showed it so beautifully well that fibroblasts and other cell types such as mesenchymal cells are so important. Fiona, thank you so much for joining us today.
Thank you. Yes, I want to echo Enrique's words. This was extraordinary. Thanks for your efforts and for sharing an example of truly outstanding translational immunology. It was remarkable. I would just like to remind people that they can ask you questions via Twitter. There's already a tweet that you can find on our Global IMMUNOTALKS account. Please reply to this tweet. And remember to use the hashtag GlobalImmuno and at Fiona Powrie. Fiona will make use of her own account to answer your questions. So thanks, everybody. Thank you, Fiona. And we look forward to next week. Thanks.
Thank you.
Bye-bye.
Bye-bye.